After learning Mendel's monohybrid and dihybrid cross and the ratio, today we will learn about the variations of Mendel's ratio. When we are talking about ratio here, we are talking about the monohybrid ratio. Though Mendel carried out the crosses with pea plants, and it is said that he was quite fortunate that he got all the ratios perfectly as 3:1. Later, experiments were conducted in similar lines, but it so happened that all the experiments conducted did not come under the ratio of 3:1. With due respect to Mendel, we still do follow those laws which are the basics of genetics, but the ratios which deviated from the normal 3:1 are called as variations of Mendel's ratio. Now, the two concepts which you have to learn as per your syllabus, that is incomplete dominance and co-dominance. These two concepts we will try to learn today. Okay, first we will go to incomplete dominance. First we will go to incomplete dominance. Now, what is incomplete dominance? It is a phenomenon in which the F1 hybrid exhibits characters intermediate of the parental genes. Now, already we have seen that Mendel's monohybrid ratio, which came to 3:1. There, the characters, the phenotypes seen were either tall or dwarf, which were the characters seen in the parents. So, even the offsprings were either tall or dwarf, accordingly whatever the parents were. The heterozygous form also showed only tall. They never showed any intermediate character. When a cross was carried out, with some other plant, then it was seen that the F1 hybrid, means the F1 individual which is always a heterozygous, it did not show the parental characters. It did not show the parental characters, but it showed an intermediate new character. That formation of new character, this process, this phenomenon, it was referred to as incomplete dominance. Now, in it, what happens? The phenotypic ratio deviates from Mendel's monohybrid ratio. Mendel's ratio was 3:1. So, it deviated from that ratio. Now, here the example taken, the example taken is of snapdragon flower. Snapdragon plants, like you all must be familiar with this type of plants. So these are the flowers, snapdragon flowers. These are also called as dog flower. Commonly, we also refer to them as dog flowers. So you can have a closer look here. You might be having them around your place also. So these were the flowers. These type of flowers were plants were taken. Sorry to say. These type of plants were taken and experiments were conducted. Those plants are also called as snapdragon, dog flower, or the scientific name is Antherhinum species. You have to remember all the terms because during your exams you might get questions related to these terms. Now, what was done during this experiment? Parents, two plants, the experimental procedure is same as Mendel had carried out. They took one plant which bore red flower of Antherhinum, another plant which bore white flowers of Antherhinum plants. Pure. They were breeding pure. Prior to the experiment. Prior to the experiment, these were also breeding pure. Then they were crossed. They were crossed together. Then let's see the genotypes. Genotypes were taken for the red flower, considering it to be a dominant trait. What was done? It was considered that red will be dominant over white. So the genotypes of red were taken as capital R, capital R. The genotypes of white were taken as small r, small r. Then the gametes procedure same. The process of carrying it out is the same. The gametes resulting out of the genotype homozygous. This is a homozygous condition. So the gametes will be same type of gametes. R and R capitals. Similarly, the white flower plants, the genotypes, okay, accordingly the gametes will be smaller and smaller. Now in the F1 generation, it was seen that the heterozygous, like previous cases, a heterozygous condition came up. One gamete from one of the parent, another gamete from the second parent. They came together forming the genotype of the offspring. This genotype, earlier case, Mendel's ratio, Mendel's cross, we saw that it expressed one of the parent's character. But in this case, quite surprisingly, what happened, it did not express either red or white, but it expressed a new color, that is pink. Here a pink flower bearing plant arose or grew out of the seeds that developed from the F1 offspring. So all the plants which grew, the F1 generations, all of them were pink flower bearing plants. Now next, they also carried out F2 cross. Now the F2 cross, how was it carried out? First, what was taken? The gametes, okay, gametes of the F1 heterozygous individual, capital R and small r, here we get. So we carry out the Punnett square like we did before, capital R, small r. These are the two gametes. This side also capital R, small r. Then the offsprings we get, when these two gametes, if they combine, possibilities are this. So what will happen? Capital R, here capital R, small r. Again another heterozygous condition, capital R, small r. And the fourth one is homozygous recessive condition. So now we put the genotypes here, capital R, capital R, small r, small r, small r. Now already it was seen that RR is red, okay, then capital R, small r is pink, small r is white. That is already in the parents. Now of course these genotypes, this is we work out later, but initially what do we see? We see only this pink colored bearing plants of F1. When the F1 were allowed to self-pollinate and self-fertilize, then it was seen that they gave rise to these plants, okay, these offsprings, they gave rise to new offsprings which were red, some were pink and some were white. The number of pinks were found to be more compared to red and white flower bearing plants. Well, upon working out the genotype, now we know how it happens and why it happens. So out of the 4 individuals possibilities, we get 1 red, 2 pinks, these 2 will be pinks, and 1 white. So ultimately what happens? What do we get? We get the ratio as, phenotypic ratio as 1:2:1. 1 red, 2 pink, 1 white. Now when we see the genotypic ratio means how many? Okay, how many this red, the genotype is this, how many of this? 1. This combination, that is homozygous condition, how many were there? Homozygous red condition, how many were there? There was only 1 out of the 4 offsprings. Then heterozygous condition, pink condition, how many were there? In this form, how many? 2. Here, I am referring to this. Okay, here is 1, here is 2 and 1. So here we see the genotype also comes to 1, RR1, capital R1, heterozygous 2 and homozygous recessive 1. So the genotypic ratio also comes to 1:2:1. Phenotypic ratio also is 1:2:1. Why it is so? Because 1 red, 2 pink and 1 white. Now when we compare with Mendel's ratio, monohybrid ratio, Mendel's monohybrid ratio, phenotypic ratio, what happened? We got 3:1. We got 3:1. 3 were tall and 1 was dwarf. In this case, what happens? We are getting 3 types of traits. Genotypic ratio in Mendel's cross, it was seen that there also you get capital T 1, capital T small t 2 and small t 1. I'm talking about Mendel's monohybrid cross. So what happens there? So there capital T 1, capital T small t 2 and small t 1. But it so happened that capital T small t, that is homozygous heterozygous condition, all came out to be tall. Okay, so that is why the phenotypic ratio was 3:1. But genotypic ratio, of course, out there also it is the same. Now here, just the explanation why it happens so. R is not completely dominant over small r. And so, heterozygous forms, that is capital R, small r, expressed an intermediate trait that is pink. Okay, so R is not completely dominant over small r, and so heterozygous forms capital R and small r expressed an intermediate trait that is pink color. So in this case, what has been seen, that capital R is not completely dominant over small r in a heterozygous condition. In contrast to Mendel's pea plants, there capital T, small t, capital T is completely suppressing the expression of the dwarf trait. But here R, the red, the allele or the factor for red is unable to completely suppress the trait for smaller. Okay, it is completely to suppress, it is unable. So what happens in this condition? Even R is expressing red. Okay, the red color is also expressed, white color is also expressed. So ultimately you get pink color. Okay, so ultimately what do we get? We get pink color. So this is incomplete dominance, where the dominant allele is not able to completely dominate the recessive allele. Recessive allele also expresses to some extent, the other allele also expresses to some extent, and consequently what happens? You get an intermediate trait. The next concept which we have to go for variation of Mendel's monohybrid ratio is co-dominance. Co-dominance means both. Okay, two types of dominance. Now what is this phenomenon? The alleles which are able to express themselves independently even when present together, heterozygous forms, are called co-dominant alleles, and the phenomenon is called codominance. Now the alleles which are able to express themselves independently. Now here alleles, now from now on we'll be using the word alleles. What are alleles? Alleles, I had already talked about these definitions in my first lecture. Okay, that is, alleles are the alternate forms of a gene. Okay, like we have seen in the previous cases, the genes for height is capital T, small t. So capital T, small t are the alleles, different forms of the genes. Mendel had called them as factors, okay, but later now we call them as genes and alleles. So here, the alleles which are able to express themselves independently even when present together. That is, if it is a heterozygous condition, both the alleles are equally expressing. The previous case of incomplete dominance, we saw that both of them are partially expressing. So we get a blending, we get a new trait. But in this case, what happens? Both the alleles equally express. So we get both the forms equally. Okay, both the traits equally expressed in the phenotype of that particular individual. This process is known as codominance. Now, the very good example of this particular phenomenon is ABO blood grouping in humans. Now, the blood grouping pattern we had already learned in class 11th. Okay, how the blood groups, they are categorized A, B, O and AB. There are 4 types of blood groups in the human population. Now we have to see here, or we will be seeing here, how actually this categorization happens. Okay, now red blood cells, that is RBC, has sugar polymers on its plasma membrane. Now we also call these sugar polymers as antigens, which we had learned in class 11th. Okay, so the plasma membrane, okay, of the RBCs, the plasma membrane, the outer surface, okay, outer layer of these cells, they have certain sugar polymers. That means some structures made of sugar polymers on its plasma membrane. Those sugar polymers are called as those sugar polymers are also called as antigens. Now these polymers, okay, when we study different blood groups, different RBCs, it has been found that there are mainly two types of sugar polymers. Some of the RBCs had one type of polymer, some had another type, okay, some have either of them, okay, some without any polymers, while some have both the polymers. So accordingly blood groups were named. Okay, if you can remember, okay, two types of antigens or sugar polymers which have been named as A and B. If they have only A type of polymer, we call them as A. If they have only B type of polymer, we call them B. If they have both A and B, we call that blood group as AB. And if they do not have any polymer, we call them as O. Now, what is responsible for it? Okay, for this. Now, these polymers, okay, now what behind every trait, every character that you see in an individual, always there are genes working. Now, even behind the formation of the sugar polymer, there is a gene I. Okay, there is this gene which is I. I is the gene responsible for formation of the sugar polymers upon the surface of RBC. Now this gene I, it has already been studied, and it has been found that this particular I gene has three forms. Okay, three forms or three alleles in entire human population. Three types. They have been named as IA, some have been named as IB, and some have been named as small I. IA, the allele, if it is present in an individual, it will be responsible for formation of sugar polymer A type. If IB is present in an individual, it will be responsible for formation of sugar polymer B type. If small I is present, then no sugar polymers are produced in that individual. But one thing to remember here is, okay, to remind yourself is that the all traits, okay, all traits or all characters in any individual is always controlled by alleles, definitely, but in that individual always there will be two of those alleles. Okay, that is the gene I is for blood. It has three different alleles in entire human population, but every individual will have any two of these alleles, of these forms. Two. Okay, we are diploid organisms, so we will have two of these alleles. Different combinations. Some may be homozygous with IA, some may be homozygous with IB, some may be homozygous with small I, or we may be heterozygous with in different combinations. Now, here as already mentioned, IA, A, IB, B, and small I, no sugar polymer. Now, let us see the how it happens. Two individuals, okay, two parents, parent 1 and parent 2. Let us say from parent 1, whatever combination of genes is present for that allele, for the gene I, maybe IA, IB, or whatever combination is there, let us say that from parent 1, one parent comes the gamete carrying IA. In the second parent, okay, in the second parent comes, say, IA, same type of allele, very much possible. Okay, from one parent comes IA, other parent also comes IA. In the offspring, what happens? That these two gametes are fusing together. So what happens? The offspring becomes IA, IA. In the offspring, these two alleles for the gene I comes together. When these two are coming together, then IA is very much responsible for formation of polymer A. So that individual, that offspring will have A type of blood group. Then let us say from parent 1, another instance, IA allele comes and from parent B, allele IB comes. When these two unite together, we get a combination IA-IB. Now, IA-IB, whenever it is present together, it has been seen that both A polymer, B polymer are formed on the plasma membrane. Consequently, the blood group, the plasma membrane shows both types of polymers or antigens, and we name them as A, B. Then, third instance, IA allele combines with small I from the second parent. We get this combination. When this combination was seen, the RBC formed A type of antigens, only A type of antigens. So, the phenotype came out to be capital A. Likewise, IB came in this combination, showed blood group B because the type of antigen formed was only B type. IB small I, it was seen the antigen formed was only B type, so the blood group came out to be B. Then in the sixth type, if one parent is giving small I, the other parent is also giving small I, combination was small I, and it was seen on the surface of the plasma membrane of RBC, no antigens being present. It was categorized as O type of blood group. Now what does it imply? Okay, what does it imply? It implies that when we come to this case, it is seen IA small I forms A. I is without any expression, small I. That means what? This is recessive gene. This is recessive allele. This is the dominant allele. Again, similar in this case, IB small I, it is seen that B type of antigens are formed, which shows that IB is dominant over small I. Small I is not allowed to express. Then coming to this, okay, third type here, you can see IA, IB, when the combination came, the phenotype was expressing both the polymers A and B. A is dominant here. We know A is already dominant. B is also dominant. So here, when they come together, these two dominants, when they come together, both expresses itself. Both are expressing, so we get both the traits in the same individual, in the same cell. This phenomenon is known as co-dominance. This phenomenon is called as co-dominance, and which was very much against Mendel's explanation. Mendel had said that in a heterozygous condition, always one will express, other will be suppressed. It is happening here. It is happening here, but not here. Okay, so this was against whatever Mendel stated. Okay, this is a variation of Mendel's statement. This phenomenon is called as co-dominance. So here we can now categorize IA homozygous dominant because they are same, both are dominant. This is co-dominant. This condition is heterozygous dominant here because it is a heterozygous condition, one expressing, other being suppressed. Homozygous dominant, again heterozygous dominant, and this is recessive. When both the recessive alleles come together, then no expression. Okay, so we get here as the recessive form. Okay, the recessive trait is expressed. Two dominant alleles, IA, IB, as I already mentioned, one recessive allele, small I. This presence of more than one allele for a particular gene is also referred to as multiple allele.